Today, we are going to talk about developmental and stress-induced programmed cell death in plants. Programmed cell death or apoptosis is a genetically programmed cellular process and a crucial component of development and defense mechanisms. Though in the plant, a true caspase system is lacking, still programmed cell death can occur throughout the life cycle at any cell type, tissue, and organ in response to a wide range of stimuli. Caspase system. Caspases actually are a family of proteases enzyme playing essential role in programmed cell death. Caspase means cysteine-dependent asparate-specific proteases. So a true caspase system is absent in plants. In plants and animals, programmed cell death plays significant role during development, structure formation, or removing unwanted tissues. It has also been reported that programmed cell death acts as a defense mechanism of an organism to prevent the pathogen spread from infected cell. A simple definition of programmed cell death. Programmed cell death is a process in which the cells voluntarily commit suicide on several occasions, among which the most prominent reasons are morphogenic or developmental changes and stress responses. In plants, the process of true apoptosis, as I already told you, is absent, but has a similar kind of programmed cell death in plants, termed as apoptotic like programmed cell death, AL-PCD, right? There are some similarities and also differences in these two processes. The general similarities between apoptosis and AL-PCD include cell shrinkage, chromatin condensation, mitochondrial permeabilization and depolarization, cytochrome C release, and vacuole leakage and fusion with plasmalemma. However, unlike animal cells, in plant cells, the content of dead cells remains in the cell itself, and there is no membrane blowing. And also, the process of phagocytosis is absent in plants, right? So, AL-PCD is morphologically and biochemically similar to apoptosis, but due to the structural and functional differences of plant cells, there are some changes in the execution process. Next is programmed cell death in plants. In general, programmed cell death is grouped into three broad categories: apoptosis, autophagy or vacuolar PCD, and necrosis. Apoptosis is an active, programmed process of autonomous cellular dismantling that avoids eliciting inflammation, right? Autophagy means self-eating. Then necrosis. When we compare this necrosis with apoptosis, necrosis is a passive process. It's a passive accidental cell death resulting from the environmental perturbation with uncontrolled release of inflammatory cellular contents, right? In plants, programmed cell death is a facet of a wide range of developmental programs, which vary from the beginning of the plant's life cycle through essential development right until the end of the plant life cycle, means senescence. So different modes of cell dying in plants are apoptotic like cell death, AL-PCD, senescence associated death, and third one is vacuole mediated cell death, which resembles autophagy, right? So like animals, when a plant cell is subjected to PCD, several changes occur. For example, as I already told you, cell shrinkage, condensation of chromatin, chromosome fragmentation, mitochondrial permeabilization, cytochrome C release, etc. Right? Now, in comparison to animal programmed cell death, the true detailed mechanism of AL-PCD in plants is unclear, right? Although presence of rigid wall associated with cell membrane prevents to form apoptotic body and lack of true caspase as well as Phagocytic cells are main reason for absence of true apoptosis in plants. But plants exhibit apoptotic-like cell death, as already we have discussed. AL-PCD that shows striking similarity to apoptosis, right? So, AL-PCD is initiated as a part of several developmental processes in response to pathogen attack and following abiotic stress. The mechanism of execution of AL-PCD is associated with activation of caspase-like molecules and the release of mitochondrial proteins, including cytochrome C. The death process is relatively quick, usually 6 hours from initiation until the final destruction of a cell. This 6-hour window allows identification of an initiation phase, a later execution phase, and a final degradation phase of the death process, which aids identification of the component of the AL-PCD death cascade in plant cells. Now listen carefully. In plants, AL-PCD is a cell death process, right? That can be defined by the presence of characteristic features like recurring corpse morphologically, where the protoplast has shrunk from cell wall, and the degradation of DNA, often, but not always, into DNA ladders, right? In plants, the role of DNA degradation is not yet clear. However, there are several interesting possibilities are there. As with animals, it may be a pathogen defense response, preventing pathogens from replicating and uh, infecting neighboring cells. Or it has been suggested that nucleases may be integral in recycling DNA, right? Phosphorus is considered to be an immobile ion in the soil, and in low phosphorus conditions it is difficult for plants to assess phosphorus without mycorrhizal association. If you remember, we have already discussed this. For plants to assess phosphorus, it's difficult without mycorrhizal association. For many plant species, nucleic acid represents an important source of phosphorus, right? Programmed cell death nucleases may therefore help to recycle DNA from dead cells by, uh, degra- by degrading it into smaller fragments that can be taken up by neighboring cells. This is very interesting, right? So, next is second type of PCD in plants, that is senescence associated programmed cell death. Senescence is a complex, highly ordered process during which plant organs undergo a series of biochemical and physiological changes. These developmental changes result in degradation of macromolecules and the recycling of their components to other parts of the plant. Senescence ends in death of the organ. However, until late in senescence, the process requires cell viability and is often reversible. When there are three positions regarding the overlap in senescence and PCD has been identified. When senescence-associated PCD was activated, it initiated a cascade of events that destroyed the cell over a relatively short period of time, 3 to 12 hours, and therefore operated under the same timeline as PCD activated by heat stress. This led to the conclusion that programmed cell death either operated as the final installment of senescence or equally plausible operated following senescence. Next is last type of programmed cell death in plants, that is vacuole mediated cell death. A large portion of the plant cell is occupied by vacuoles. You know this. And the presence of lytic vacuoles plays a major role in plant cell death. This type of cell death is known as vacuolar cell death, right? Plant cells contain two types of vacuoles in different types of cells, in tissues, lytic vacuoles and protein storage vacuoles. Protein storage vacuoles may contain many kinds of proteins, especially defense and stress related, and the pH value found close to neutral, right? So, lytic vacuoles helps in ion and water homeostasis of the cell. The mechanism involved in vacuolar cell death involves the increase in the size of the vacuoles by fusing small vacuoles and result in the formation of large vacuoles. Another important topic related to programmed cell death is plant programmed cell death in response to biotic and abiotic stress. Plant programmed cell death occurs mainly during the time of plant pathogen interaction as well as in response to different biotic stresses. Plant pathogen interaction mediated hypersensitivity response is a mode of broad spectrum resistance in plants. Whether the pathogen is compatible or incompatible is determined by the interaction between resistant genes in the host and the avirulence genes in the pathogen. Hypersensitive cell death is a localized rapid death of the cells at the point of infection, and it serves not only to restrict the growth but also stop the invasion of pathogens to other cells or tissues of the plant by undergoing suicide of the infected cells. The complex signaling of these hypersensitive reactions involves a cascade of events such as protein phosphorylation, reactive oxygen species production, and modification of ion flux. Right? Hypersensitive hypersensitivity responses actually is a defense mechanism against biotrophic plant pathogens or microbes, which depends on nutrients for survival on the host. However, it has been observed that hypersensitivity response might be beneficial in the early stages for local adaptation against the infection, but not in the later stages. All hypersensitivity responses against all these HR induced means hypersensitivity response induced PCD have have a feature common which distinguishes it from vacuolar type. That is, this type of PCT is not initiated by vacuolar swelling and release of lytic enzymes inside plant cell, right? So this is all about plant programmed cell death. Thanks for watching. If you like this video, make sure to subscribe for more. Thank you.